Hello everyone, welcome to Ritika's online teaching academy. In previous lecture we have discussed about the C3 cycle. In today's lecture I am going to talk about the C4 cycle. So those who do not have not subscribed till yet, please subscribe. So in order to get more videos which would be helpful for you in NEET exams. Okay, now I'm going to sharing my screen with you. Okay, so today's is my fifth lecture and it is on C4 pathway or C4 cycle. Okay, C4 pathway occurs in the plants which are found in hot and dry tropical regions such as maize and sugarcane, sorghum, etc. So they have developed different kind of mechanism for the fixation of CO2. We have seen in C3 plants that they have ribisco enzyme which fixes carbon dioxide. But the plants such as maize and sugarcane which follow C4 pathway have different mechanism of fixation for CO2. Now, as these plants are found in hot and dry tropical regions, the stomata of the leaves of these plants remain closed or partially open. As we all know that plants which are found in hot and dry climates, if their stomata remain open, then what will happen? Water vapor will go out by the process of transpiration and the plant will become dry. So in order to save that water loss, these plants' stomatas remain closed or remain partially open, and in this way they reduce the loss of water vapor. But also, this results in the less supply of carbon dioxide. So, if there is less supply of carbon dioxide, then how they are going to make the fixation for CO2? So there is a different way of fixing carbon dioxide prior to entering into C3 cycle in these plants. In this way, we understand that the plants which follows the C4 pathway or C4 cycle also have C3 cycle in them. So it means that C3 cycle is found almost in all plants. So before going or entering into the C3 cycle, there is a different way for that. Now we will look for this. So as we all know, ribisco stands for ribulose 1,5-bisphosphate carboxylase oxygenase enzyme, and this is the world's most abundant enzyme. And we have already discussed about it in C3 cycle. Now I am recalling it. So this enzyme have active sites which can bind to both carbon dioxide as well as oxygen. Means it has affinity for both CO2 as well as O2. Since it is an enzyme and we know that enzyme binds with its substrate at its active site. So it has greater affinity for CO2. When CO2 and O2 are nearly equal, generally it remains equal in atmosphere. So it means that in that case, they will have greater affinity for carbon dioxide. Hence the photosynthesis process goes on. But sometimes what happens, this CO2 and O2 ratio disturbs. In that case, ribisco, this enzyme, instead of binding with carbon dioxide, it binds with oxygen. Means in this time, it is working as a ribulose 1,5-bisphosphate oxygenase, not carboxylase. So its oxygenase activity will help it to bind with oxygen instead of carbon dioxide. And when CO2 is there in more quantity, then its carboxylase activity will come into play. So what will happen if it binds with oxygen? It will lead to photorespiration, and this process is of no use. And also this occurs at the expense of ATP. So energy is also going wasted, as well as the process is not useful for the plant. So in this way, we can say that it is a side reaction of the photosynthesis or of the carbon dioxide fixation in this plant. So we can say that this process, photorespiration process is a wasteful process. But this happens in C3 plants only. It means that C3 plants efficiency or are less efficient in comparison to C4 plants. We will study it further. Now, in C3 plants, Rubisco enzyme, which has greater affinity for both CO2 and oxygen, carry out photorespiration to some extent. While in C4 plants, photosynthesis reduces photorespiration by concentrating more carbon dioxide around Rubisco. So this is one advantage of C4 plants in photosynthesis, that they do not allow photorespiration to go inside them. And how they do it? By concentrating more carbon dioxide around the Rubisco enzyme. Means Rubisco is also found in C4 plants, but in the C3 cycle. It means that C3 cycle also is a part of the C4 plants. So how it is achieved? It is achieved by a special type of leaf anatomy of C4 plants. Means the thing which is responsible for the reduction of photorespiration is a special type of anatomy of leaf which are found in C4 plants. So in order to tolerate higher temperatures, C4 plants have different types of leaf anatomy, and this is one of the advantage why photorespiration does not occur in these plants. In this way, it will lead to the higher yield in C4 plants as compared to the yield in C3 plants. So once the CO2 concentration is high in the atmosphere, C3 cycle occurs very well. But in C4 plants, even at less concentration of CO2, the photosynthesis goes on very well. So a special type of leaf anatomy found in C4 plants is also known as crans anatomy. Crans meaning wreath, wreath meaning rings, means there is a ring-like structure, or this structure consists of large cells which are also known as bundle sheath cells, and they surrounds the vascular bundles. So what are the characteristics of these bundle sheath cells? These bundle sheath cells have thick walls which are impervious to gaseous exchange and have no intercellular spaces. So this quality of bundle sheath cell would not allow carbon dioxide to escape from it. So in this way, they are concentrating more and more carbon dioxide around the Rubisco enzyme. So these cells may be arranged in several layers around the vascular bundles. Means there is a ring-like structure and there may be several rings of bundle sheath cells in order to reduce the loss, or you can say in order to concentrate more CO2 around the Rubisco enzyme. So we can see in this picture, it is a leaf anatomy of C4 plant where between the upper and lower epidermis, between the upper and lower epidermis, there are mesophyll cells rings and bundle sheath cell ring is located. So this bundle sheath cell or ring of bundle sheath cell is covering the vein, or you can say vascular bundles. And around these bundle sheath cells, a layer of mesophyll cells is present. The number of chloroplasts are more in comparison to number of chloroplasts in mesophyll cells in. So we can say that there are more number of chloroplasts in bundle sheath cells as compared to those of mesophyll cells. Here you can see that stomata is present and it generally remains closed or partially open in these C4 plants. And we know that stomata are responsible for the gaseous exchange. It means whatever the gas, oxygen and carbon dioxide, its movement inside and outside depends upon the stomata opening and closing. So now the Hash-Slack pathway. So before discussing about the Hash-Slack pathway, I will give you a brief history about it. Hash and Slack was a scientist and they did the similar kind of experiment as the Kelvin did. He performed with plants, he subjected some plants to the radioactive 14 carbon dioxide, and then he analyzed the metabolites obtained from those plants after photosynthesis. And what he found, that the first intermediate product which was found in those plants is um, that C4 acid, oxaloacetic acid. So it means that in this case, something instead of that PGA, we are getting oxaloacetic acid, which is not a 3-carbon compound, as we can recall that PGA is a 3-carbon compound, while oxaloacetic acid is C4 compound or C4 acid, which is found in C4 plants. Now, the other thing which he noticed, that the carbon dioxide fixation, or you can say the photosynthesis process, is generally divided between the two isolated compartments. One is in mesophyll cell and the other one is bundle sheath cells. Okay, so mesophyll cells, as we have seen, that these form a ring-like structure around the bundle sheath cells, and bundle sheath cells form a ring-like structure around the vascular bundles. So what processes which are differentiated among these two types of cells we will discuss now. So in mesophyll cells, you can see mesophyll cell, the upper one is mesophyll cell and the lower one is bundle sheath cell. So in mesophyll cell, when from the stomata carbon dioxide from the atmosphere comes, it converts into the bicarbonate ion, which is HCO3 minus. Another molecule which is found inside the mesophyll cell is PEP. PEP stands for phosphoenol pyruvate. This PEP combines with HCO3 minus in the presence of PEP carboxylase, or we can say phosphoenol pyruvate carboxylase enzyme, and fixes carbon dioxide into a C4 acid. And this C4 acid stands for oxaloacetic acid. At this point, this OAA either as such, or it may get converted into malic acid or aspartic acid. This malic acid, or we can say it as malate also, aspartic acid as aspartate, which are 4-carbon compound, and OAA is also 4-carbon compound. So these acids, either malic acid or aspartate acid, get transported into the bundle sheath cell through the plasmodesmata. There is a channel between mesophyll and the bundle sheath cell, which is called as a plasmodesmata, for the conduction or transportation. So this C4 acid gets decarboxylated. Decarboxylated means removal of carbon dioxide from that C4 acid. So this removed CO2 now enters into the C3 cycle, or it gets fixed by the C3 cycle. So we can say that here C3 cycle is occurring in the bundle sheath cell instead of the mesophyll cells. While in C3 cycle, we have seen earlier that this fixation of carbon dioxide or photosynthesis occurs in mesophyll cells, but here it is in bundle sheath cell. So this is one of the difference between the C4 plants and the C3 plants. Now after decarboxylation, this C4 acid gets converted into the C3 acid. This C3 acid is generally pyruvate. This C3 acid now gets transported back into the mesophyll cell. Here this C3 acid or pyruvate regenerates the PEP. So in this way, PEP means phosphoenol pyruvate. In this way, C4 cycle gets completed. So this pathway was given by Hatch and Slack. That's why it is called as the Hatch and Slack pathway. So we understood the whole mechanism here. Now with the help of a flow chart, it will get more clear. So SCO3 minus ions, which we get from the carbon dioxide, once carbon dioxide enters into the mesophyll cells, it gets combined with the phosphoenol pyruvate, which is a 3-carbon compound. In the presence of PEP carboxylase or PEP case, phosphoenol pyruvate carboxylase, it gets converted into the oxaloacetate or oxaloacetic acid, which is a 4-carbon compound. It may get converted into the malate or aspartate, which is also 4-carbon compound. Then it enters into the bundle sheath cell. And when it enters into the bundle sheath cell, it gets decarboxylated and removes one carbon dioxide. And this carbon dioxide is fed into the Calvin cycle. And malate itself gets converted into the 3-carbon compound, which is pyruvate. What it means that this 4-carbon compound, after the removal of one carbon in the form of carbon dioxide, how many carbons are now remaining in that? 3 carbon. And this 3-carbon acid is called as the pyruvate or pyruvic acid. This pyruvate again gets transported back into the mesophyll cell, where it converts into the PEP or phosphoenol pyruvate, which is a 3-carbon compound again. In this way, it completes the C4 cycle. And at this step, ATP molecule is also used. ATP gets converted into ADP, an inorganic phosphate. So this ATP does the function of phosphorylation of this pyruvate to make it converted into the PEP phosphoenol pyruvate. And this process occurs in the presence of an enzyme called as pyruvate orthophosphate dikinase. In short form, we can say it as PPDK. So we can say there are some differences between the C4 and C3 cycle. First is, the important thing is that C3 cycle is common to all plants. It means it occurs in all the plants. But in some plants, there are some adaptations in order to, or you can say, in order to save them or make them adaptable to their environmental conditions. So as we have seen in C4 plants, leaf anatomy itself is very beneficial for those plants to function or to carry out the photosynthesis process even at the very low concentration of carbon dioxide. While the disadvantage in C3 plants is that there is ribisco enzyme which is found, which has both the carboxylase and oxygenase activity, means it has affinity for both CO2 as well as O2. It may carry out a side reaction of oxygenation and may lead to a wasteful process called as photorespiration. While that photorespiration is not a disadvantage for the C, means it does not occur in the C4 plants due to their special type of leaf anatomy. The other thing is that ribisco enzyme, in the case of C3 plants, it is present in the mesophyll cells where it carries out the C3 cycle. While in the case of the C4 plants, we have seen that Calvin cycle occurs in the bundle sheath cells, not in the mesophyll cells. So in bundle sheath cells, Calvin cycle is carried out. Okay. The other advantage of C4 plants is that there the enzyme which is found is PEP carboxylase, which fixes carbon dioxide, and it has only the carboxylase activity. It does not have the oxygenase activity. This is also one of the reasons why photorespiration does not occur in the C4 plants. Okay, so these are all about the today's lecture. In the next lecture, I will discuss about the uh, SCAM plants, or you can say crystallization acid metabolism process. So in order to get more videos like this, please do like, share and subscribe my channel. Thank you.